Welcome to our video about RNA polymerase II structure and function on behalf of MCDB 427 at the University of Michigan. Eukaryotes have three types of RNA polymerase. Each one transcribes different types of RNA in the eukaryotic cell. Today, we will be focusing on RNA polymerase II, which transcribes mRNA and most siRNA. RNA polymerase II has 12 subunits encoded by the Pol-R family of genes. They are named RNA polymerase B, RPB, and are named in order of size from largest to smallest, meaning RPB1 subunit is the largest one. RNA polymerase II can be compared structurally to bacterial core RNA polymerase, which is composed of subunits beta, beta prime, 2 alphas, and omega. The 2 alpha subunits can be compared to RPB3 and RPB11, beta prime to RPB1, beta to RPB2, and omega to RPB6. For now, we're only going to talk about RPB1. When looking at RPB1 separate from the rest of polymerase for a moment, we can see that the carboxy terminal domain is composed of a long string of heptad repeats, meaning 7 amino acids that repeat 52 times in vertebrates. Here, we have drawn one example of that heptad sequence, but keep in mind that it is repeating throughout the C-terminal domain region. These heptad sequences are composed of many serines, threonines, and tyrosines, which are able to be phosphorylated. This phosphorylation pattern can vary between the heptad repeats, meaning the amino acids that are phosphorylated are not always the same. For the sake of simplicity, in this example we are only going to show phosphorylation of these two serines. However, the other heptads and the phosphorylation states of their respective amino acids are not shown. Phosphorylation of an RNA polymerase subunit is significant and can account for its interaction with the DNA or specific proteins during transcription initiation, elongation of an mRNA, or during RNA processing. RPB1 is heterogeneous, meaning it exists in multiple forms, or in this case, different phosphorylation states. The study that first characterized RPB1 isolated the RNA polymerase II subunits from a mouse tumor called a plastocytoma and then ran them on an SDS-PAGE gel. Here is an image of the real gel that they ran with all of the RNA polymerase II subunits. What was surprising was that the top 3 bands were all supposed to be RPB1. This indicated that there are 3 RPB1 isoforms, which they named 2O, 2A, and 2B. 2O is a twice phosphorylated 2A. 2B, however, is missing the repeating heptad sequence normally found on the CTD region of RPB1. Researchers confirmed this by using antibodies against that sequence, which only bound to subunit 2A, but not to 2B. The conclusion they proposed was that there was a proteolytic enzyme that cut off the repeated sequence, in turn converting 2A to 2B. This is why 2B is so much smaller than either 2A or 2O. 2B is simply a degraded product, meaning that it only exists in a test tube. Incubating 2O with phosphatase removes the phosphate groups and reverts it back to 2A. This shows that the phosphorylation of 2A leads to the creation of 2O, and the opposite is true to reverse back to 2A. 2O can also be converted to 2B when a protease removes the C-terminal domain with the heptad repeats. Knowing that there are different forms of the RPB1 subunit leads to the understanding that there are different versions of RPB1. These forms might have different purposes, such as 2A binding the promoter, and 2O, the phosphorylated form, being responsible for elongation. When thinking about the larger picture and how to visualize RNA polymerase II as a whole, scientists struggle to come up with a clear picture because of the differences in structures of RNA polymerase across species. Not all the subunits are essential for normal transcription. Some RNA polymerase lack RPB4 and RPB7, but can transcribe just fine. The heterogeneity of the RNA polymerase II complicated the usual process of X-ray crystallography to visualize the structure as a whole. If there was a mixture of the proteins, some lacking RPB4 and 7 and some having RPB4 and 7, crystals could not easily form. Roger Kornberg was able to resolve this problem by designing a mutant yeast polymerase that completely lacks the RPB4 and RPB7 subunits. This was useful because then he could successfully use X-ray crystallography to give us an understanding of how the different subunits interact with one another and what the structure looks like as a whole. RPB3 initiates RNA polymerase II assembly. After subunit synthesis, 3 joins with 2, and then 1, allowing 5 and 7 to enter. 5 recruits 6 and 8, and then 3 brings 10, 11, and 12. 4 and 9 come fashionably late, added last to the growing complex. Going back to our stereo view of RNA polymerase II, we can note that in this model we are missing RPB4 and 7 as well as the CTD region of RPB1. You can still see 3 distinctive features of RNA polymerase II from this image: the cleft, the clamp, and the jaws. The cleft is this slit here. This is the region of RNA polymerase II that binds to the DNA template strand. The slit fits the single strand of partially unwound DNA. It contains a magnesium ion at its active site, deep in the base of that cleft, that acts as a cofactor to stabilize DNA binding. Next, we have the jaws. The jaws are positioned at the opening of the cleft. RPB1 and RPB9 form the upper jaw, and RPB5 forms the lower jaw. Lastly comes the clamp. When the RNA polymerase is not attached to the DNA, the clamp is open, allowing access to the active site. Once the elongation complex forms, the DNA is in the active site and the clamp closes. With transcription underway, the clamp must stay closed over the DNA so the polymerase doesn't fall off too early before the transcript is complete. The clamp is lined with basic amino acids, which sharply contrasts the rest of the enzyme, which has acidic amino acids only. This design strengthens the binding to DNA, which has mostly acidic residues. We hope you've enjoyed watching our video on behalf of MCDB 427 at the University of Michigan. Thanks for watching, and go Blue!